Hi, it's great to be here. We've had some incredible talks today. The amazing complexity of biology has been sort of unleashed upon us this morning. And maybe I wanted to take a slightly different tack now. And rather than starting with something really complex, I wanted to start with something really simple. As the complexity of a problem increases, the number of people who can solve that problem decreases. Okay, it's pretty obvious, right? But let's take that to its logical conclusion. As we continue to ratchet up the complexity of the problem, the number of people who can solve it globally, I'm talking about all 8 billion people on the planet, drops off to 0. And it's at that point when nobody can solve problems because they've got so complex that we've hit up against something that we term the cognition barrier. And the cognition barrier is a massive issue, not just for me, but for you and everybody else on the planet. And the reason for that is the problems that we need to solve most, those that are most impactful in terms of our health, our prosperity, and perhaps even the survival of the species, they sit on this side of the cognition barrier, the side that we can't solve. And so the biggest challenge that we face here today is how do we break through the cognition barrier? And to do that, we actually need to reimagine what the scientific discovery process is and what it could be. And I think retrospectively, we'll almost look back at this point in history almost as a point in which humans transition from being central to the scientific discovery process to being integral to, but not essential to it. And the work that we do at LabGenius is all about reinventing the scientific discovery process so we can autonomize the discovery of novel therapeutic proteins to treat people. Now, this idea that we can autonomize the scientific discovery process, it's not new. People have been dreaming of building a robot scientist, autonomous systems that can do science for generations. But actually, now is the time in which these novel ways, these novel smart robotic platforms that are able to run scientific processes are actually coming online. Now, I'm going to talk a little bit more around how these work and how we're using them. So the basic architecture for these systems is pretty well established, and it's actually very intuitive. And it's actually very similar to the way in which we do science today. So in this scientific discovery process, you start off by generating a hypothesis. And if you're a human scientist, of course, that happens up here in your head. And then you test that hypothesis. You test it physically in the real world. And again, if you're a human scientist, maybe you do that with your hands. But with a system like this, the hypothesis generation happens in the world of bits on a computer, and the hypothesis testing happens in the real world, but rather than being done with your hands, you can do it with robots. Now, the real challenge when it comes to building a system like this is that you've got to really tightly integrate the world of bits and the world of atoms, so information can flow seamlessly between those two worlds. And that's a real challenge. And in my view, that has been the limiting factor, that integration between these two worlds, that has stymied the development of platforms like this. Now, in the context of the drug discovery process, we can now start to go a little bit more deeper, a little bit more granular, and this is the way in which it works. So we're in the business of defining new therapeutic molecules, and the way in which that process runs is computationally, we'll design a set of molecules. Those sets of molecules will then be fabricated. They'll be made here in the real world, in the world of atoms. They then get tested, and then the information, that characterization data, is fed back into the world of bits to build computational models that are descriptive of the solution landscape for that particular molecular property or set of molecular properties. And then you can co-optimize new molecules, and you can go round and round this cycle and searching through solution space until you find molecules that have the requisite properties. And as I mentioned, the big challenge of building these sorts of systems is that you have to integrate the world of atoms and bits in order to pursue this ML-driven active learning process, you have to be able to run around this loop exceptionally quickly. And that's not only an infrastructure challenge, that's also an organizational challenge. And if we could jump to the next video, please. So what you see here is the infrastructure that we've got set up just down the road, in fact, 2 tube stops away down in Bermondsey. So in an old biscuit factory, we knocked down several walls, and we kitted out this biscuit factory with all of the equipment required to run the experimental processes that are involved in testing thousands of new proteins. And I'm going to go into a little bit of detail now into how the system works and why it's so complex. So when you come up with that panel of designs, you have hundreds of DNA sequences that the computer generates and says, right, here are DNA sequences that encode protein therapeutics that we believe will be high performing. The next step there is that you actually got to make those. You've got to make those in the world of atoms. So we use a mass modular cloning strategy to assemble all of the DNA in parallel. So we then have all of these DNA sequences. They've got to go into a bacteria to be amplified up, to be increased. You then got to purify those. You then have to sequence those, and we do that in-house with nanopore sequencing, so we can get that sequencing turnaround in a couple of hours. And then once we've validated that all of the sequences are correct, those sequences, those DNA sequences, then have to go into a mammalian cell so that they can be produced at high levels. You then have to purify the protein, because these are all protein therapeutics, antibody therapeutics, out of a mammalian cell. And it's only then, once you've got that to that stage in the process, that you can actually functionally characterize them. And the functional characterization then happens across a suite of disease-relevant functional assays, as well as biochemical, biophysical developability assays. So you've got this massive infrastructure and this very long workflow that you have to go through, and you have to seamlessly integrate all of those different pieces, so you have the information flowing through it in a highly automated way. And really the challenge here is how do you run around that loop incredibly quickly at high throughput and generating data that's both really clean, but also you can control for the inherent biological noise in the system. And that's not only a technical challenge, but that's a massive organizational engineering challenge. Because whilst the argument here is, humans are no longer central to the discovery process, as I mentioned, they're incredibly integral to it. So actually building these platforms takes a very multidisciplinary team of exceptional scientists and engineers spanning all different disciplines, from data science to software engineering to protein science to cell biology, synthetic biology, etc. And bringing all of those folks together to work together to build out one of these platforms. Back to the slides, please. Okay, so you've heard a little bit around, I guess, the concept behind one of these systems and the kind of infrastructure that you have to set up in order to affect it. But what are we actually using it for? Well, at LabGenius, one of the problems that we're trying to solve is how do we treat cancer better? And the way in which we're doing that is we want to encode logic into protein therapeutics so that they can selectively distinguish between a healthy and a disease cell, and then selectively kill that disease cell. Now, that's something that's incredibly hard to do. But what we found is by running this active learning process and letting the data guide us through the sequence space, what you end up with is being pulled into areas of design space that you would never have explored as a human protein engineer. So you get these quite unusual designs that, again, don't really make sense to us as humans, but they make sense to the underlying models that have designed them. And we find that these are massively improved with respect to the performance. So we found molecules that have a killing selectivity of over 400 times competitor molecules or benchmark molecules that are currently in clinical trials. These results really are incredible and testament to the power of this underlying process. So what I'm going to do is jump now to the next video and show you this process in action. Okay, so here are the molecules that we're working with. These are the proteins that we're engineering with that system that you saw on the previous video. In this case, this is an example of an immune cell engager. So that's a particular type of protein that's able to recruit one of your white blood cells and use it to kill a cancer cell. And it's made up of different pieces. One piece to bind to a cancer cell, a couple of other pieces to bind to a cancer cell. So obviously, here's in a tumor. If you zoom in to the tumor, what you'll see is a cancer cell, and every cancer cell on its surface has a unique postcode, a unique set of cellular receptors. And the molecule's able to dock here, as well as being able to dock onto the immune cell. So it can bind to both the immune cell and also onto the cancer. And then it's able to bring those two cells together and catalyze the killing of the cancer cell. Okay, and the challenge, this is not a new form of molecule, by the way, but the challenge with the conventional immune cell engagers that operate like this is they really struggle to differentiate between what's a healthy cell and what's a disease cell. And the net result of that is you get killing of both your healthy cell and disease cells. And if you're a patient, that means you either don't get the treatment because it never got through clinical trials, or you do get the treatment and you have to deal with all of the toxicity events that go with this. Now, using this active learning process, we can search into areas of solution space, in areas of design space, where these molecules are able to distinguish between the healthy and the disease cells. So we're able to optimize them in a way that you can't using these conventional methods. And with these optimized immune cell engagers, you can distinguish between healthy and disease cell. And the challenge here is one of the levers that you're pulling against is the affinity, the binding affinity of the molecule. You bind too tightly, you kill everything. Equally, if you bind too loosely, you don't bind to anything and you kill nothing. So here you're titrating or managing the binding affinity of these molecules in conjunction with a whole range of other features that you're simultaneously engineering in to ensure that these molecules are not only highly selective for your cancer cell, but additionally, they're highly active and highly developable. So it's that co-optimization challenge that we're solving here. So we're building computational models across all of those different features in parallel, optimizing across them, using this active learning process to create very novel, non-intuitive designs. Okay, so you've seen the architecture of this system, some of the infrastructure that we've had to build out to create it, and an example of one of these applications for it. But maybe I just want to end on a broader thought here. The reason I'm really excited is systems like this that you see here have the potential to transform the whole of the way in which we discover drugs today. And not only that, this can be applied to the discovery of whole other areas of science and technology. So I'm really excited about this next step that we're taking in reimagining the scientific discovery process and the implications that will have for the future prosperity of humans. So with that, I'd just like to thank everybody and also make a small plug for a panel discussion that's happening later today with some fantastic panelists at 2 p.m. on the future of computational drug discovery. So if you are free and interested in that as a topic, please do join us then. Thank you very much for your attention.
Thank you very much, James. First question, I caught part of your presentation backstage and was listening to the phrase, and I couldn't get it out of my mind, where you were saying something along the lines of human beings are no longer central to this process. Yet you also mentioned the multidisciplinarity of the teams that are needed to get your vision going. So can you talk us through that tension of, are human beings becoming more important, less important, or just differently important?
Yeah, it's a great question. In my view, this is almost like a Galilean moment for science, in the sense that pre-Galileo, or maybe pre-Copernicus, people thought, obviously, the sun went around the Earth. Obviously, not so much. It's the same in science, in the sense that by reimagining the scientific process, what that will enable us to do is to solve harder problems. But humans are still absolutely integral to that. So you can't build these systems or set up the problem spaces without having incredibly talented teams, multidisciplinary teams of people actually building the platforms. But the platforms themselves operate in a way that's quite independent to the traditional scientific discovery process. And that creates some interesting conversations and tensions in the sense that when protein engineers join LabGenius, it often takes them a while to get used to the fact that the designs of these molecules are actually computationally driven. And you look at these molecules, and then you try and post-rationalize, okay, it looks like this, maybe this is the mechanism of action. But the reality is, unpicking the molecule's mechanisms of action, it may not be something that you can actually understand as a human. So I think it's causing us to redefine our relationship with science and the scientific process.
And does this give you a new opinion about the nature of creativity?
Yeah, so what's really, I guess, important to say is that these systems have an exceptionally narrow intelligence, so they're really good at solving very tightly defined problems. And I think the really creative aspect that you see of certainly the team at LabGenius is actually how do you set up these systems to be able to solve those problems? Because one of the most important pieces is, yes, the system can look in the solution space, this design space, and find high-performing molecules. But it's the protein engineers and the data scientists who sit down at the beginning of a program and actually demarcate the area of design space that's going to be searched. So I think it's a new form of creativity, a new application, and hopefully a more interesting one as well, in the sense that through the heavy use of automation, a lot of scientists don't actually want to be conducting experiments day in, day out, and they're using a different part of their brain.
One of the questions that I've been hearing from some people in the audience is something around the nature of talent. And as we're getting ready to train people to do the kind of work that you're doing, the sort of future of drug discovery, et cetera, should people have a core function? Or are you leaning potentially towards something that's more multidisciplinary, more polymath approach to knowledge, different skills? Like, what are you looking for when you're building your teams?
Yeah, so I think you absolutely do require a core function. We have functional teams across everywhere of the business, and within a functional team, everybody will be, say, an expert in data science or cell biology, etc. But what's absolutely essential is that everybody within those functional teams is intellectually curious, and that they're able to at least understand at a superficial level what's going across on in the other teams. And one of the constant bits of feedback I get from folks at the business is it's that interdisciplinary collaboration and discussion that is driving the creativity and innovation.
And are you seeing that talent pool here in the UK sufficiently, or are you having to tap the global talent pool?
I mean, we're really fortunate in the sense that some of our best hires have come internationally. But I would say it's maybe less around the lack of the scientific depth, but more the advantage of bringing in folks who have had experience of the startup ecosystem before and the biotech ecosystem before, in the sense that within other geographies, that's a lot more mature than it is, say here in London.
Right. What are the blockers that are stopping you from, at the moment? Is it just time or something else that's stopping you from realizing your vision fully? Is there anything that, if there are people here in the audience who could wave magic wands that could help you, what would you say, I need to remove X, Y, and Z, and then we can go for it?
Okay, so this is a really boring answer, but whenever a minister asks me this question, I think this is actually the most impactful thing, which is in the UK, the transport system doesn't work at all. And like, it sounds like a really kind of...
Hold on, are you saying leaves on the line are keeping you from...
It's too hot, it's too cold.
Okay.
It's the wrong day of the week.
So transport infrastructure.
But I'm genuinely serious. When you have a very multidisciplinary team where folks are coming in from Oxford, Cambridge, London, etc. If you don't have a good transport ecosystem, that is genuinely an issue. I know it sounds like super boring and people want some like whizzy bang answer, but that's...
How do you feel about crumbling concrete or childcare?
I also have opinions on those. Yeah.
I thought you might, which is actually really good though, because often when we have these discussions, we can think it's about money or something very ephemeral, and in fact we need the trains to run on time. So for is it Grant? Who's the Minister of Transport these days? They change so quickly.
I asked the Shadow Science Minister, and the answer was, it's not simple. So
it's not simple. Yeah. It's true. And if you could describe your vision of what success looks like for you and how you're going to know when you get there, tell us a happy story.
So for us, the big moment of success came when we were able to really see in the data, when we were running multiple cycles of this process, these molecules were improving cycle after cycle in a really systematic way. So from the kind of the deep science, molecular biology, synthetic biology, data science perspective, that was a real win for us. The next point is seeing these molecules progress into later stage parts of the discovery pipeline and ultimately into patients. That's everything that the team is excited about.
And how far are you from it, do you think?
Yeah, we're still a little bit away from that, but I think we'll get there over the next couple of years.
Amazing. I'm going to let you off a little bit early, just so that you don't feel pressured to say something in 20 seconds, unless you wanted to say, if you could tell the people in the audience to do one thing to learn about this space, what should it be?
Yeah, I think if you want to learn about the space, probably the best thing to do is acquaint yourself with the deepest problems that need to be solved. I feel like there's a big danger in over-indexing on the importance of one technology or another, but it's actually the real problems that have to be solved that should drive the innovation. So I would look at it like that.
Thank you so much. Dr. James Field.
Thanks.
Cheers.